Okay, so today I'll be talking on microbes and the environmental role of microorganisms in the natural system. That is how these microbes play an important role, significant role in the environment to survive. In this lecture, we'll discuss about the biochemical cycling, carbon cycle, nitrogen cycle, phosphorus cycle, and in next lecture, we'll be talking on sulfur, iron, and manganese cycle, mercury cycle. Now, these cycles are very important for environment to survive in different living and non-living organisms to establish like a symbiosis between these two systems. So in my first lecture, we'll be talking on biochemical cycling. Now in biochemical cycling, the sum of the microorganism or the microbial world, physical, chemical processes that drive the flow of element between the sediment water and the atmosphere is known as a biochemical cycle. Now in this table, if you look carefully, you will find this is cycle that is called biochemical cycle for the carbon cycle. In carbon cycle, you see the reduction form is methane, CH4, and then yes, this is the significant gaseous component present in this. Now, intermediate oxidation state form, which is the major form of the of this cycle that is being converted into different product, is the carbon monoxide. Then we have a oxidized form is CO2, carbon dioxide. Now, similarly, you have a nitrogen, sulfur, and others. So, in case of nitrogen, we have ammonia, ammonium organic nitrogen, then we have a nitrogen gas, that is N2, and then nitrous oxide and nitrite and nitrate. So, the conversion of these different states of the carbon compounds, nitrogen compound and the sulfur compound. For example, in case of a sulfur, we have a hydrogen sulfide, H2S, then HS group in organic matters, and then this is called elemental sulfur, that is S0, and this is called thiosulfate, where we have S2O3 O with 2 minus valency, then we have a sulfide, then sulfate. So all these intermediate are all this compound which are so single element, single molecule, that is carbon, nitrogen, sulfur, ferrous, they are converting their product in the presence of environmental condition, in the presence of oxygen, in the presence of CO2. And how do they convert, and what is the utility and significance of these compound, like nitrogen compound, then we have a sulfur compound, then CO, carbon monoxide, carbon dioxide, every compound, organic or inorganic compound which are formed through these elements during the cycle is impacting a lot on the environment. Now, if you look at this complicated redox potential pH, where we talk about the redox potential, then we talk about the pH, oxygen concentration, influence the availability of the terminal electron, etc. How the conversion of these molecules takes place. So, what is the chemistry behind that, right? So, to understand the chemistry behind that, this like a depth, it's not in a scale. This is basically showing the manganese, ferrous, and then hydrogen sulfide, then nitrites, and how the aerobic and anaerobic state, that is oxic or anoxic states, behave like in the presence of oxygen or in the absence of oxygen. So, there are two states of these conversion, one we call oxidized state and reduced state. So, the reduction reaction and oxidation reaction taking place. So, this is for example denitrification. So, denitrification is this is the delta G minus 800 kilojoule per reaction. So, this is the nitrites, and this is nitrate. Denitrification takes place. The product of the denitrification is CO2, and then you have equal balance of these amount of CO2 release and nitrogen, hydrogen oxide, that is water, and OH ion. So, this reaction is basically is outcome and product of the nitrification or denitrification. Similarly, you have a reduction reaction takes place. So, this is denitrification. Now, we have reduction reaction. In the reduction reaction, we have again the manganese oxide reduced, and this manganese oxide reduction takes place which reduced carbonate ions, then manganese and hydroxyl ion. This is called reduction. Similarly, you have a reduction of ferrous, reduction of sulfate. We will discuss this in the later slides. So, the important point here to understand how these reduction and oxidation reaction takes place in the presence of oxygen and in the absence of oxygen, and this reaction taking place. Now, how these reactions are basically impacting on the environment. So, this is all environmental gaseous system where involved in the cycle. Now, just look at the carbon cycle, attributes of carbon cycles. The carbon cycle is building block of life and found everywhere in the earth. So, everywhere on the earth and globe, the carbon cycle is present. Now, it exists in different form. For example, reduced form of carbon like methane, organic matters like enriched form, energy form, oxidized form of carbon monoxide, CO, carbon dioxide, CO2. These are oxidized form of this carbon. Then we have no, the oxidized form and reduced form means like electron transfer. It is all the game of electron. The chemistry behind that is how electron transfer taking place. So, accordingly, you have presence of hydrogen ion or loss of hydrogen ion, or the hydrogen ion being removed by the oxygen ion. Now, oxidized form like carbon monoxide, CO, carbon dioxide, energy poor form, then flux is a movement of a carbon between the reservoirs, that is we call the sources and the sink, and the cycle begins with the carbon fixations and converting the CO2 into organic compound. Now, it looks like we are talking about the chemistry. It is not a chemistry, but it is a chemistry which is going on in the microorganisms. So, it means the microorganisms are basically involved in the reduction and oxidation process in the presence of environment, or when they are present in the environmental conditions, different environmental condition. Now, look at this cycle, that is a carbon cycle, where we talk about the fossil fuels, that is combustion. So, the CO, carbon monoxide. So, this is the red one is oxidative. Here this line is oxidative reductions or oxidation taking place where the CO2 is formed. So, there is the addition of oxygen. So, this is methano and methylotrophy process we call, where the methane is converting in CO2 through oxidation process, whereas this is called reduction process, where the CO2 is converting into the methane. So, this is similarly, if you talk about the other way, that the CO2 is converting into organic matter. So, this is called CO2 fixation, or we call the photo and on chemotherapy. So, this is reduction process, or we call it reductive phenomenon, whereas on the other side the organic matter is converting into CO2, and when the organic matter is converting into CO2, it is called respiration and fermentation process. This is called oxidative reduction neutral. So, the oxidation reduction process are basically continuous cycling process of these elements which are present in the environment, and these microbes play an important role in doing so, and this reaction is also going on in the microorganisms as well. Now, the microbial carbon fixation, how the carbon is fixed and why the carbon fixation is required. So, microbes fix at least 50% of the earth carbon is fixed by the microorganisms, and the key player for the fixation of a carbon is marine photosynthetic bacteria, and then we have Prochlorococcus, and we have a Sanicococcus, Prochlorococcus, Sanicococcus, then Protist and Diatoms. So, all these organisms are involved in the fixation of the carbon on the earth, that is 50% carbon on the earth has been fixed by these microorganisms. Anoxic environment means like microbes fix carbon without oxygen. So, even if oxygen is not present, as you have seen in the earlier cycle, for example, here the CO2 is present, so we got oxidation process and to form CO2, but I think even without anoxic conditions also we can do, the microorganisms can work well and they can form the microbes fixation of a carbon dioxide, carbon. The anoxygenic photosynthesis where the no oxygen is released. Similarly, chemolithotrophy. In chemolithotrophy, which use inorganic chemical instead of light, right? It's not autotrophy. It's not like it's a chemolithotrophy. And deep ocean microbes are oceanic microorganisms or microbial flora fix many of these carbon sources through these reactions. Now, carbon transformations and methane. How the carbon transformations and methane takes place, because methane gas is very important for environment, and the environment produces a lot of methanes through carbon cycle. The fixation of a carbon, that is carbon organic matter pool, where the organic matter is converted into like again reduce through the CO2. So, the carbon can return to CO2. So, it is like oxidation of this organic matter in the presence of oxygen that is carbon is converted into CO2 carbon dioxide. So, this is like cycle of formation of from carbon to carbon dioxide, from carbon dioxide to organic matters and so on. So, this cycle is beneficial for the various environmental condition. Now, aerobic respiration is like using oxygen. So, like we respire, we are breathing, so we need oxygen. If there is no oxygen, we cannot breathe. So, respiration is not possible without the oxygen, because all the reaction in the mitochondria and oxidation reaction is taking place in the presence of oxygen. So, oxygen is required for the living organisms to live. Now, anaerobic respiration is called fermentation, which basically do not require any kind of oxygen or oxygen product or oxygen molecule. So, anaerobic reduction is produces the methane gas CH4 from the carbon without any oxygen. Only archaea can produce methane from O2, from hydrogen oxide or H2O, or from H2 hydrogen. So, hydrogen plus CO2 or H2 plus acetate, they form the methane in the case of the archaea bacteria. So, archaea bacteria can form these. Now, methane oxidation is aerobic, like protobacteria. We have alpha protobacteria and gamma protobacteria. So, anaerobic archaea or bacteria denitrification takes place in this case. Now, aerobic methane oxidation, methano and methanotrophic protobacteria, where the CH4, O2 and CO2, 2H2O, they form the reaction. So, aerobic methane oxidation, methanes and methanotrophic protobacteria are required for that, where the methane is converted into CO2 and water molecule in the presence of oxygen. Similarly, another class of bacteria we call methylo mirabilis. So, methylo mirabilis is basically oxyphera. This is again the methane is converted into CO2. Methane is converted into CO2 through the same reaction in the presence of nitrate. And then methanogenesis is called in archaea bacteria, it is methanogenesis, where the CO2 is converted into methane directly, and carbonate ions, bicarbonate ions convert into methane and produce the CO2 again. So, the cycle of CO2 is again the CO2 is converting this, and then again CO2 is being produced. So, cycle is like this going on. Now, similarly in archaea we have reverse methanogenesis. Reverse methanogenesis, archaea and SRB, we have a methane plus sulfate, and again bicarbonate ions are produced, then we have H2O is produced, and so this methane is utilized here. So, this cycle is going on, methane is produced. So, the whole methanogenesis cycle is produced. So, methanogenesis and methylotrophy is basically methane is produced by the archaea bacteria, terrestrial and marine anoxic sediments, right, and it diffuses the upward, where the oxidized by one of the three genes like methanotroph and only discovered protobacterium, then we have candidatus, then methylo mirabilis, oxyphera. So, all these bacteria are basically involved in production of these methane methane gas, where the carbon molecule is completely recycled in the cycle. Now, the major methane sources, ruminant gut is a methane source. Then you have various sediments like rice paddies, then we have coal mines, sewage treatment plant and landfills. So, all these are the sources for the methane production, and the microorganisms they are present in these sites and to produce the methane. Then wetlands, we have marshes, mangroves, swamps, they all are rich of methane producing sites. Archaea in termites gut, right. So, archaea can also produce the methane in the termite guts. The key microbes like methanogen produce methane, then methanotroph, they consume the methane. So there are two group of bacteria. One is methanogens bacteria, which basically help in production of the methane, and another is methanotrophs, which basically help in consumption of the methane, which are so this cycle of methane production like CH4 production and CH4 consumption. So this is how the methanotroph, right, they are consuming this, right, and this is how the methane is formed, this methanogens. So this kind of a two group of bacteria, they are basically producing the methane, and they are also uh, consuming the methane in the environment. So by this phenomenon, that is that by production and by consumption of the methane, you can maintain the balance in the environment and the cycle is complete. What is lignin resistant? Lignin resistant compounds, lignin is basically a tough polymer in plant cell wall. So plant cell wall, the covering of the plant cell wall is a thick polymer, right? And it degraded by fungi, streptomyces. It need oxygen, break down lignin efficiently. And Rhodopseudomonas, this is called Rhodopseudomonas species, and this is a specific like Palestris species, which required the uh, anaerobic degradation. It's a very slow process, and slow breakdown lead to accumulation of these lignin, and then peat block the formation. So this is basically the lignin we call the resistant compound, where the lignin resistant compounds are produced and doesn't allow the formation of these accumulation takes place. Now mineralizations and nutrient cycle. Uh, Mineralization, as we all know, that the deposition of the mineral, right, where the minerals are deposited. So mineralization means organic matter. Simple inorganic compound like CO2, NH3, CH4 and O2, they all are produced, right, and nutrients recycling depends on the relative abundance. If one nutrient is abundant but another is limiting, the excess may not be used. For example, the protein chitin provides more nitrogen than can be used relative to available carbon dioxide. So this is one of the example. I mean, so you can have a protein chitin which provides the more nitrogen, because protein has a lot of nitrogen molecule, and they can use for relative to and available for the carbon cycle. So how the mineralizations and nutrient cycle continuously working in the presence of these molecules, these compounds, CO2, ammonia, methane, hydrogen, and nutrients converted to biomass become the temporarily immobilized. So the nutrients which are converting into the biomass, like for example biomass of the plants, the biomass of animal biomass, so they become temporarily immobilized. Now the macronutrients limitation. The microbes using the carbon rich substrates need other elements like N, P, S, E, like nitrogen, phosphorus, sulfur, and ferrous. Now the growth limitation occur if any required nutrients is scarce. So how the growth is limited? So this is called the growth limiting factors. If any nutrient is, for example, if nitrogen is not present, phosphorus is not present, sulfur is not present, iron is not present, then there is a growth is limited. So in many organisms, both in plant and living organisms in animals, the growth limiting factors are these nutrients, which are very important. We call the macronutrients. Now, and the cycle we call the carbon cycle or nitrogen cycle or sulfur cycle, they are basically responsible for providing these macronutrients to the environment, maybe plant, maybe to the animals. Lederberg, this is Liebig's law of minimum. This is one of the scientists, Liebig, who derived a law and called it the Liebig law of minimum. So that is, what is the minimum quantity of these macronutrients are required. So growth is controlled by the scarcity of the nutrients, basically limiting factor for the growth of this. So there is some limit. What is the minimum quantity of these nutrients which limit the growth. I mean, if you provide cross the limit of these nutrients, that is more nutrients are being provided, then the growth can be again attained. Open ocean microbes are often nitrogen limited. So if there are no nitrogen, they cannot produce their energy, or they cannot live, or they cannot survive. Now, if you look this complex organic structure, substrate characteristics that influences the decomposition of and the degradability. Now, the decomposition and degradability of this biomass, whether it is a plant biomass or whether it is a, like we call composite, or we call them as animal biomass, where degraded. Now, this decomposition or degradation of these living organisms basically produce the these elements which present in large quantity. Sometimes the degradation takes place in the presence of oxygen, sometimes in absence of oxygen taking place. So it depends on what kind of compounds are formed. So for example, if you take a substrate like a starch, so basic subunit is a glucose, and the linkage of these glucose is alpha 1,4 linkage. Now this is the orientation and chemistry behind the the glucose molecules which linked to form a starch molecule, because glucose is a basic subunit. So number of n number of glucose form one starch molecule. So more the glucose in the chain, more strong the starch, or larger size of the starch is formed. So the linkage is alpha 1,4 and alpha 1,6 linkage. The element present is all carbon, hydrogen, oxygen. Nitrogen and phosphorus is not present, and both with and without presence of oxygen, it get degraded. Now, if you want to degrade the cellulose, which is again a very important substances, again it is made of a glucose, but the cellulose is a beta 1,4 glucose linkage. There's a difference between cellulose and starch. And again, it has carbon, hydrogen, oxygen, and it can also degrade in the presence and absence of oxygen. Now, if you talk about the hemicellulose, hemicellulose is a C6 and C5 macro monosaccharides, right? So both C6 and carbon 6 and carbon 5 pentose and hexose monosaccharides. It has beta 1,4 and beta 1,3 and beta 1,6 linkages, and again they are carbon, hydrogen, and oxygen present in that, and both in the presence and absence of oxygen it get degraded. Lignin is phenylpropene as a subunit, and this phenylpropene are linked with the carbon-carbon and carbon-oxygen bonds. They are again having the carbon, hydrogen, and oxygen, and also the phosphorus, because it is a propylene, propene, and then it can also with and without or even absence of oxygen, they can work well. They can degrade it. Chitin is a N-acetyl glucosamine. N-acetyl glucosamine is a membrane linkage, where beta 1,4 linkage is formed with N-acetyl glucosamine, and these N-acetyl glucosamine, large number of subunits of N-acetyl glucosamine are like linked concatemers, and then they form a chitin with the beta 1,4 linkage. So it has all carbon, hydrogen, oxygen, nitrogen, but not phosphorus, and then we have both in an absence and presence of oxygen, it can degrade it. Hydrocarbons are aliphatic and cyclic aromatic compounds. So these aliphatic compounds are cyclic compounds. They are basically they don't have a bind, they have their own chemistry, and it has carbon and hydrogen compounds and present, and they can be degraded both in an presence and absence of the hydrogen and oxygen compound, oxygen molecule. The lipids are the glycerol fatty acids. Some contain the phosphate and nitrogen, so it has all like carbon, hydrogen, nitrogen, phosphorus, and they can degrade it in presence of oxygen and in the absence of oxygen. Both oxidative, both like in anoxic and oxic conditions, it can degrade it. The nucleic acid is a purine and pyrimidine base is sugar phosphate present, so it is a phosphodiester linkage takes place, and glycosidic linkage bond takes place. Hydrogen bonds are there, so they have all hydrogen, nitrogen, oxygen, phosphorus, and it can be degraded in both presence and absence of the. Now, nitrogen cycle is it exists in redox states from -3 to +5. right? And then depending on oxidation state, so nitrogen species can act as electron acceptor, example nitrate like NO3 and NO2 in anaerobic condition, that is in the absence of the oxygen. An electron donor, example nitrites, NO2, ammonium, NH4, and in chemolithotrophy condition. So this is the condition, that is a state where the chemolithotrophy in how electron donor in the chemolithotrophy state, and in anaerobic respiration we have electron acceptor for this. The key principle is fully oxidized species NO3 can only accept electron, fully reduced species NH4 can only donate electron, nitrite can do both. So this is how the nitrogen cycle work. Like so, it can form different nitrate compound. Redox reaction takes place, and it can at one state they donate electron, and another state they can accept the electron. Now this is a schematic presentation of this cycle. That is where the, if you look the organic on the top, there is the organic matter, there is amino acid or amino and nucleic acid, and this amino and nucleic acid is basically decaying taking place by, we call it mineralization. So many genera are involved in that. So ammonium oxide is formed, ammonium is formed, then this oxidation of this NH4 ion is taking place. This is called nitrification step. So nitrification step is nitrite is formed, and the example is nitrosomonas and nitrosococcus, some chronoketose bacteria are formed. Now this major part is the microbiome is nitrosomonas and nitrosococcus, which are involved in the formation of nitrate. Now this nitrate and nitrite conversion is taking, we call the dissimilarity nitrite, where the reduction of nitrate takes place. Now it serve as a terminal electron acceptor during the anaerobic respiration, when gas N2 and NO2 are produced and released into the atmosphere, process called the denitrification. This is called the denitrification process. Now similarly, this oxidative state further convert the nitrite to nitrate, so we call the nitrification step. In nitrification step, second step, the NO2 serve as electron donor, as we told earlier, and in the case of chemolithotrophy, and nitrobacter and nitrococcus are the bacteria which are involved in the conversion of the nitrite to nitrite. Now assimilatory reaction where the nitrite reduction takes place, where the nitrite reduction takes place, serve as a nitrogen resources. So this nitrate serve as a nitrogen resource, and many genera of the bacteria of the group of Nitrococcus and Nitrobacter, they are involved in the conversion of this into organic nitrogen. So finally, the whole cycle, that is the decomposition of the nitrite nitrogen, organic nitrogen, decompose it from plant source or from animal source to convert to ammonia, to nitrite, to nitrate, and then again nitrate is converted to the organic compound. So the whole process of this involved in decaying, oxidation process, nitrification process, and nitrification is step 1 from ammonia to nitrite, and from nitrite to nitrate. This process of oxidation, 2 oxidative processes involved. So this complete the whole cycle. Now the bacteria which are involved in the whole cycle is Azotobacter, Clostridium, Rhizobium, and many of the bacterium, they are involved in the nitrogen fixation, and this nitrogen fixation is involved in the various leguminous plant. So conversion of N2 gas organic. So by definition, we can define as a nitrogen fixation as a conversion of a nitrogen gas into organic nitrogen and amino acid, purine, pyrimidine, and then performed only by certain bacteria and archaea, as we have shown here. So this nitrogen which is formed from O2, this is basically N2 like this, and then N2 is formed nitrogen fixed by the organic matter, and this organic matter is then with the help of several bacteria, they can form organic nitrogen in the form of ammonium, ammonia, nitrites and nitrates, and again from nitrate is recycled into the organic compound, organic nitrogen. The oxygen sensitivity is also important. The nitrogenase enzyme, that is the nitrogenase which involved in the formation of nitrogen. In O2 sensitive, but fixation occurs under both oxic and anoxic conditions. So these are the nitrogen fixing microorganisms, where the aerobic nitrogen fixations, where we have Azotobacter species. So some of the microorganisms, they are present in the presence of oxygen, and some they in the absence of oxygen. So aerobic, that is in the presence of oxygen, which convert the nitrogen, which fix the nitrogen, is azobacter species, and cyanobacteria trichodesmium species. Sorry. Cyanobacteria and trichodesmium species and azobacter species are involved in the nitrogen fixation of the aerobic condition, that is in the presence of oxygen. In anaerobic nitrogen fixation, Clostridium species are involved, where the oxygen is not required, because most of the Clostridium species like Clostridium perfringens, which cause the gas gangrene in various infections, they are always grow in the absence of oxygen. So therefore, always the wounds are keep open so that oxygen can be supplied and Clostridium growth may not takes place. That's a point here. The symbiotic nitrogen fixation is a leguminous plant. So there's a symbiosis between legume symbiont like plants, rhizobia like alpha protobacteria, some like beta protobacteria are also present. Then actinomycetes, the Frankia species, woody shrubs we call, then heterocysts forming cyanobacteria, Anabina species for azolla water ferns. So this is how these different plants are associated symbiotically with these bacteria, which are helping in the fixation of nitrogen, and these plants require the nitrogen basically. So this is the ammonia incorporated into the proteins, nucleic acid, and other biomolecules. The ammonium and nitrification, the organic nitrogen ammonium NH4 through dissimilation and mineralization, and ammonium is electron donor only in the cumulotrophic condition like nitrite convert into the nitrate. NH4 to nitrite ammonia, this is called ammonia oxidation. Now nitrification are nitrifying microorganisms like ammonia oxidizers, which help in oxidization of ammonia, as Nitrosomonas and Nitrosococcus, the mesophile archaea, marine ammonia oxidizer. So these are the important microorganisms which maybe. So to remember all these name of these microorganisms linked with their associated plant for the nitrogen fixation is not easy, but I think you can remember some of the most important events like ammonia oxidations and nitrite oxidation is very, very important from various perspectives. Sometimes the questions may come in the net examination also. You can also go through these to remember the nitrosomonas, the role of nitrosomonas, or the presence of nitrosomonas in conversion of ammonia, nitrite, and nitrite to nitrate. So all these things to be remembered, I think is very important. Then mesophilic archaea, marine ammonia oxidizers, right? Then nitrite oxidizers are nitrobacter species, right? Related bacteria. Some Nitrosomonas eutropha is perform anaerobic, like in absence of oxygen. The ammonia formations and ammonia oxidation takes place. Now, how it reduce? The reduction of nitrate is basically can use in assimilatory nitrite reductions reduce and then incorporate it into the biomass, that is again organic biomass, and dissimilatory nitrite reduce using the terminal electron acceptor using the anaerobic respiration. Now this is the dissimilatory nitrate reducers. Now Geobacter metallireducens are basically involved in dissimilatory reactions, as in conversion, dissimilation of this nitrate compound or nitrate molecule, which reduces us basically. Desulfovibrio, again it's archaea. Desulfovibrio species are also present. Denitrifier producers producing N2 and NO2, Pseudomonas, denitrification takes place. Now greenhouse gases like N2O produced during the ammonia and oxidation, nitrifications and denitrification, especially by marine ammonia and oxidized archaea. This is called an ammox reaction. An ammox reaction is basically anaerobic ammonia oxidation. This is called shortcut of nitrogen, and ammonia convert to nitrite to nitrogen. So this process is called ammox reactions and performed by the the planktomycetes and cumulotrophic reaction, where the nitrites convert to again the nitrate by we call nitrite oxidation process, where no light is required. Now what is the significance of this is it removes the ammonia from the environment without passing through nitrate. So it is important in oceans and wastewater treatment process. So this is how the NMOX reactions is very important. Now you understand the whole cycling process of these compound carbon, nitrogen cycles are involved in environment to play a very important role to make the environment clean and to provide the survival for various lives in the environment. Now this is all about. Now we have another lectures on some other compounds and the cycles soon. Thank you very much.